Okay, well, good morning. Thank you everyone for being here in person. I am so happy to see you. And I think that deserves a round of applause. Yes, absolutely. And we have many of our alumni from around the world watching us right now. So thank you to all of you for also participating. Before we begin, I just wanted to acknowledge that the University of Manitoba campuses are located on the original lands of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. It's very important that we always start our events that way. It's a really special part of what we do here at the university. So if this is your first time being at a Learning for Life, welcome. We're happy that you're here. If you participated virtually, again, welcome. And if you've been here with our Seniors Alumni Program, which I recognize some of you, which was pre-pandemic in person, thank you for being here. We really appreciate your support. Over the last 2 years during the pandemic, over 4,000 alumni and friends participated in our virtual Learning for Life program, which is amazing. They came from 20 plus countries were joining us. And so it was usually from my dining room table is where you saw me on the screen. But it was really great that we were able to engage with our alumni that way. And our researchers were just more than happy to participate in those sessions. So thank you for being here. So it's also homecoming week. So happy homecoming. I hope if all of you drove through Chancellor Matheson to get here, you saw the homecoming banners. There's so much that's going on over these 4 days, September 21st to September 24th. Today's Friday, the second last day of homecoming. Tomorrow, let's cross your fingers there is no rain, but there's 2 Bison games tomorrow. Tickets are free. The football game's at 2 p.m. Kickoff is at 2, tailgate is at 1. And then our women's soccer team is playing at 7:30. Is kickoff, tailgate is 6:30. Again, as I said, tickets are free for everyone. If you're interested in going, you can go to our website to grab tickets there. But take advantage of as many things that are going on during homecoming during this time. So another thing that happened during homecoming is last night we had a pretty special event called the Distinguished Alumni Awards. And I am so delighted that one of our recipients is our speaker today. It's wonderful because he's from Montreal, and so for him to be able to be here and speak to all of us today is really special. So I'm going to save the introduction of our speaker to our Dean from the Faculty of Science. But before I do get to that, just a few housekeeping details. The washrooms are quite literally just outside of this room. So feel free to go there anytime during the lecture. There's also, after the lecture, there's coffee, tea, cookies, water, that kind of thing. There's actually some right now. So again, refill your glasses if you'd like to get some. After the lecture, there'll be a Q&A session, and we're taking questions both virtually and from everybody in person here. So I'll have a mic roaming around, and then one of my colleagues will be looking at the questions from our participants virtually. So hopefully, so please ask lots and lots of questions. I'm sure Peter's very open to them, so please do. Okay, so with that, I'm going to turn it over to Dr. Brian Mark, who is our Dean of Faculty of Science, to introduce our speaker today.
Good morning, everyone. Thank you, Tracy. It's a pleasure to be here. It's actually wonderful to be back in person on campus. If you have a chance to walk around campus, all the students are back. It's really nice for us to have a normal fall term after 2 years of being away in the pandemic. So it's my pleasure today to introduce Dr. Peter McPherson, the James McGill Professor of Neurology, Neurosurgery, Anatomy and Cell Biology at McGill University, and he's also the director of the Neurodegenerative Disease Research Group. Last evening, Dr. McPherson was awarded the 2022 University of Manitoba Distinguished Alumni Award in Advanced Innovation at the Celebration Excellence Gala that's held annually by the university, which was a really wonderful event. Dr. McPherson is a 2-time graduate of the University of Manitoba. He has his Bachelor of Science degree in Zoology in 1986 from the Faculty of Science, and his Master of Science degree in physiology from downtown under the supervision of Gary Cheng in 1988. He then went on to receive his PhD in neuroscience at the University of Iowa College of Medicine under the supervision of Dr. Kevin Campbell. Dr. McPherson is internationally renowned for his work on neurology, neuronal cell biology, and membrane trafficking that occurs within these cells. So this is a phenomenon that, among other things, transports and localizes proteins within the cell so they can carry out their functions in the regions of the cell where they're needed. He continues to make seminal contributions to this field, and he recently published papers describing how SARS-2 gains entry into cells by clathrin-mediated endocytosis, which is a protein-driven membrane trafficking phenomenon. His work has also identified how dysfunction of basic cellular systems can cause Parkinson's disease and insights into how Parkinson's disease progresses with age. Dr. McPherson is widely published, and his research has graced the covers of numerous prestigious journals, including Nature magazine. So for those of you who aren't aware, for a scientist to grace the cover of Nature magazine is like a musician gracing the cover of Rolling Stone magazine. You know, it really is a remarkable tribute to the broad impact that one's research has on their particular field. It reflects that and is in keeping with the many invitations that Dr. McPherson has received across the globe to present his remarkable scientific findings. So today, Dr. McPherson will be presenting a talk entitled "The Making of an Unhealthy Brain and the Tools Required to Understand Why It is Unhealthy." So with that, I'll pass it over to Dr. McPherson. Thank you for joining us.
Thank you. Well, thank you very much, Brian, for that kind introduction, and to Tracy and the organizing. Like Tracy organized this talk. She organized the homecoming, and she organized the alumni event last night, which was really tremendous. It was a lot of fun. It was, you know, she said it was going to be like the Academy Awards. I thought, yeah, sure it isn't, but it really was. It was a lot of fun. So thank you. So we all perceive from in here. Okay, everything we perceive in the world is in here. We see, we hear, we smell, we feel. But what are we perceiving, and how do we perceive it? So if you prick your finger, every human being in the world will tell you that the pain is in your finger. But is it? Okay, because if I soak your finger in some ice cold water, or I treat you with Novocaine and I block the conduction from your finger to your brain, you won't feel the pain. So is the pain in your finger, or is it in your brain? And of course, the pain is in your brain. But your brain is really good at doing something. It's good at tricking you. Your brain tricks you all the time. It makes you feel like the pain's in your finger because that's where the problem is. You can't pull your brain away because that won't help. Pull your finger away. Okay, and so the brain is amazing. I'm going to give you a couple of examples about perception. And I want to do this because I want you to understand how important it is that the brain perceives the world. And then I want to tell you about an example from my own work where that perception is lost because of a very subtle change in the human genome that leads to a very severe problem in brain development. What about color? Okay, is color out here, or is color in here? So is my backpack red? Is my shirt black? Is that green? Okay. So is the color out here in the world, or is it here in my brain? So I'm going to give you a couple. This is just for fun. Okay. So what I want you to do, and I hope this will work, if we could turn the lights down just a little bit out front, that would be possible, because it would be better. Okay. So maybe not. Okay. Hopefully this will work. What I want you to do is I want you to stare at the black dot. Just humor me on this one. There we go. That's much better. Thank you. We can turn it up later. Stare at the black dot. Just focus on the black dot. Don't look at me. Don't look at your friends. And spend 30 seconds absolutely focused on that black dot. And I'm going to quietly count to 30. And when I get to about 5, I'm going to start counting out loud. And then I'm going to switch the slides. And I'm going to ask you what you see. Okay, so go. Stare at the black dot. 7, 6, 5, 4, 3, 2, 1. Okay, what did you see? Did anyone see a castle with a blue sky? Some green grass? Yeah, but the picture's undeniably black and white. Right? So where did the blue sky and the green grass come from? Okay, so what it actually turns out, you can turn the lights back up now, thanks. What lands in your eye is not color. What lands in your eye is electromagnetic radiation of specific wavelengths. Color is created in your brain. Okay, the photoreceptors in your eye are energized by those certain wavelengths of light. They convert it into electrochemical energy. It transmits a signal into your brain. And so the perception of color is in your brain. Okay? Let's do one more. And this is a lot of fun. And my brothers are here and they introduced me to all this old rock and roll. This is Led Zeppelin. This crowd is of an age, I think, mostly that maybe the young guys won't know who Led Zeppelin is, but okay. So what I'm gonna do is I'm gonna play you about a 40-second piece of music. And I use this as an example of how good your brain is at integrating things, integrating color, integrating feel, integrating touch, memories, everything, and putting them all together. Okay, so are we ready with the volume? Okay, so here we go. So I'm gonna play about a 40-second song, 40-second clip. Plan. Try again. Now what I'm going to do is I'm going to play the exact same bit of music, but it's going to be played backwards. Okay, and at the time this song came out, there was a lot of this going on, right? I buried Paul in the middle of strawberry fields, right? When you played it backwards, people were looking for messages, and strikingly, they were never looking for messages that were positive. They were always looking for evil, satanic kinds of messages. So we're going to play this backwards. Did anybody hear any words in that?
Yeah, Satan.
Satan. Anyone hear Satan? One word, Satan? Okay, now let me tell you how your brain, show you how your brain, and if I give you just the right amount of information here, by activating another part of your cortex, let's try it again. This exact same clip. Yeah, it was there, right? It's amazing that once you know what you're supposed to be listening to, you can hear it. This is the same phenomenon when you're sitting in a restaurant and you hear some music playing in the background and you're not paying attention. Suddenly you remember the song, and now you're, without paying any more attention, you suddenly know all the lyrics to that song. So what's going on in all of these examples is it's your brain. See, and there's a very younger version of somebody I know holding a brain in my lab. It was a demonstration we were doing. And this is the amazing thing about the brain, is it's able to take all this information coming in, visual, audio, sensory, all this stuff, and integrate it seamlessly. And so, you integrated all your brains integrated the words I put up in front of you now with the sound, and you just heard it all perfectly clear. And that's your brain seamlessly doing this. Okay. So how does the brain accomplish all of these tasks? And actually, sadly, I would say for neuroscientists, we don't actually know. No one has a really good model of how brain turns into mind. how brain turns into cognition. You know, the brain's an incredibly complex organ, that's for sure. And it's got an incredibly complex cellular architecture. And the one thing I'll tell you though is it's very energetic. So your brain is about 2.5 kilos, say. So that represents about 2 to 3% of your body weight, maybe 4%, something. It uses about 20% of all your cardiac output. Every time your heart pumps, 20% of all that blood goes to feed your brain. And this is whether you're taking a test, out for a run, or sleeping, right? Your brain is a very demanding organ in terms of the amount of blood that it uses up, in terms of the amount of energy it takes. So the brain has a multitude of cell types. This was drawn by my daughter, Rebecca, I think maybe when she was 12 or 13. So the brain's got different cell types, but we're just going to talk about the neurons, okay? So this is an example. I really like it. It's hung in my office, of course, for many years. So here's two neurons connecting to each other. And so here's the cell body of one neuron. There's the nucleus, and she drew all the nice dendrites. And then here's the axon. The axon's leading away, okay? And the axon then starts to form synapses with the next cell through these processes where the axon connects to the dendrite of the next cell. And that's a synapse. And it's really at the synapse where the processing, where the information processing takes place, okay? And there are, it's estimated, so there's about 80 billion neurons in your brain. And it's estimated that they form about 1 × 10 to the 15 synapses. That's 1,000 trillion synapses, okay? So, and it's through these complex synaptic, through these complex synapses and the network properties that these neurons form that your brain can do that it can do the things that we're doing. And these are really complex cells. So you have a cell type in your spinal cord called a motor neuron, and I'll come back to that because there's going to be a little thing about ALS in the second bit of the talk. So the cell body lives in your spinal cord, and it's 100 micrometers in diameter, 1/10 of a millimeter in diameter, and it can send out a process that goes down to my toe a meter away. Okay, so let me put this into perspective. If we blew the cell body up to the size of this room, then the axon would leave that cell body, and the axon would be about as wide as the door. The synapse would be on the border with Saskatchewan. Okay, that's real. That's the dimensions of this thing. So these are incredibly complex architecture. And the proteins have to move from part A to part B of these cells, and that's, as Brian alluded to, that's what we do. We try to understand these kinds of processes. So here's the networks that form, very complex. This is just a nice view. And here's what happened. Here's what a normal human being brain looks like. Okay, so this is a brain. This is an MRI. So here's the cortex, complicated. Here's where most of the 80 billion neurons are in a human being, at least. And this structure here is called the corpus callosum, the one here in white. So we're looking at the side of a brain. This is called a sagittal section. Here's the brain stem, the spinal cord leading down. Okay? And this corpus callosum contains about 300 million axons that connect the two hemispheres of the brain. And here's some structures I want to point out here in dark. They're black. These are what are called the ventricles. These are each different ventricles, and they're all sort of connected. This is part of the circulation in the brain. There's a circulation in the brain, and it cushions the brain. It provides immune cells. It provides nutrient exchange. Okay, so here's a normal human brain, and here's what happens in a disease brain, going back to the title. So you don't need to be a neurologist or a neuroradiologist to look at these MRIs and understand that there is something severely wrong with these brains. When we first saw these images, here's the cortex. We thought maybe there was something wrong with the MRI, but no, this is cortical atrophy. This is of about a 2-year-old child who basically has such an underdeveloped cortex. Here's the corpus callosum, which that's what it looked like in the normal brain. Here's what it looks like in this disease brain, okay? And here's those ventricles, massively swollen, massively overextended, okay? So what these two individuals have is a disease that my lab discovered called DEND5A epileptic encephalopathy. So Each of the individuals has a one nucleotide change in their genome. And that one nucleotide change in their genome leads to a one amino acid change in one protein called ND5A. And that one amino acid change of that 1,500 amino acid protein, which is one of 20,000 proteins expressed, causes this. Okay. So one of the questions is how come this doesn't happen more often, right? I mean, it's incredibly complex, this development. So these poor patients. So anyways, I'm gonna. So the protein is called ND5A. This is a picture of the protein, just a model of it. It has this protein module here called a den domain and other bits, and none of this is too relevant for this audience. Okay. But here's now that neuron again, a little more stylized. And what we think this protein does is it functions in a pathway that carries proteins from a structure called the Golgi to endosomes and to the surface. Okay, and again, the details of this aren't too important for the point of this talk. Okay, but if you've got 20,000 proteins expressed in a cell, they have to be expressed. They have to be transported to the right place in the cell. That's where they function. They have to be expressed at the right level. They have to be turned over, made anew constantly. And these kinds of trafficking processes are critical for this. Okay. So we had discovered this protein in my lab, this ND5A protein. We discovered it through a long history of work that my lab had done, and very exciting stuff. And we were trying to just figure out what cell biologists do, neurocell biologists like me. People are interested in trying to understand how the subcellular aspects of the cell, within the cell, how does that relate to the formation of the neurons, to the connectivity that leads to the brain and mind. We're down at that cellular level, right? So when we try to figure out the function of this protein, we want to know what cells it's expressed in, where it's expressed, where it's localized, all those kinds of things. So this is a little bit of data. Don't worry about it too much, but it's called a Western blot. And we use an antibody against this protein, and we test what tissues it's expressed in. So you can see just by the band here, by this dark staining, it's expressed in the brain and expressed in the spinal cord, but it's not expressed in all of the non-neurological tissues. And if we look at tissues during development, we see that this ND5A protein is expressed early in embryonic brain, but as you get to more of an adult brain, and these are rats, there's much less expression. So we had published bits of this in an abstract form, and we were contacted by this guy, Burj Manassan, at Sick Kids in Toronto. And Burj had a family that he'd been following in Saudi Arabia. So this is the pedigree of the family that Burj had been following. So they had 7 children, 5 of them were, 3 boys, 2 girls. The circles are girls, the squares are boys. And perfectly normal, healthy kids, but number 6 and 7, that's those 2. Okay, so they had a homozygous recessive mutation in a particular amino acid, D173 proline. It was a 1 amino acid change in these 2 girls, and that led to this phenotype. And you can see, and those were the MRIs I showed you. Those 2 MRIs I showed you were of these 2 girls. And then we had subsequently found another girl in Jordan. Her sister had died shortly after birth, but that's her, so that represents her. And she has another mutation, single amino acid mutation. So these were just 2 patients that we'd found. And this work was done by Chan Shui Huan, and here was our former prime minister who visited our lab during the election campaign. Chan Shui was wearing an I Am Canadian t-shirt. She was Chinese, and she was trying to get her landed immigrant status. So she thought if she wore her I Am Canada t-shirt when Harper came by, that would help. It did not. Anyway, so we published this original work in a paper in the American Journal of Human Genetics in 2016. And that's now where a new graduate student comes in named Emily Banks. And this is work that's ongoing and that we've submitted for publication just recently. So at the time, we only had 2 patients. And then another paper came out and revealed that there was 2 more with these mutations. And so what Emily wanted to do was see if we could build a cohort of individuals. So what happened is shortly after we published our paper, physicians started to contact us because they had children that had some kind of neurodevelopmental disorder. But they didn't know what it was, so they sequenced the child and they sequenced the parents, and they saw a mutation, a change of a variant in this DND5A gene. Then they went into the literature, saw our paper, and called us or emailed us. There's also public databases. So Emily learned how to go through those public databases, and we found more variants, and you can see it's building. So, and then there was word of mouth between our colleagues and, and other than their colleagues. So in the end, we now have 15 patients from 14 families with 21 mutations. So this is a very rare disease, okay? It's a very rare disease, but often what happens with these rare diseases is, you learn a lot about the mutations, you learn a lot about the function of the protein, and you learn a lot about brain development because the patients have phenotypes, right? And I'm sorry, I forgot to go through them. They have severe intellectual impairment, not surprising given the MRIs that you saw. They have, so intellectual impairment, basically depending on the patient, they can't speak, they can't understand language, they can't perceive. I introduced this lecture with perception. They don't perceive the way you and I perceive. They can't sit up. They can't roll over on their own, okay? These children are absolutely dependent on caregivers. And they have epileptic seizures. They have what are called coarse facial features, and they have ventriculomegaly, that big swelling of those ventricles. So what we wanted to figure out now that we had a larger patient base was, is there a DND5A common disease? Because what happens in any one of these individuals, they have a variant in DND5A, but they have other variants in their genome. So Emily developed this phenotypic questionnaire where we asked physicians about the behavioral aspects of their individuals, their neurological and their non-neurological phenotypes or symptoms. She put about 150 questions on this, and a really nice study in that she had to contact all these physicians and geneticists and get their consent, and it all has to be done in a certain way with ethical approval. And all that information comes back, and we tried to interpret it. And basically what we found was that of the 15 patients, so one patient didn't have any of the symptoms that we put on the questionnaire, okay, we asked about. So we're not even sure if that patient is an ND5A, but of the 15 patients, 14 have global developmental delay, meaning they are intellectually impaired. Although it turns out there's really a range of that impairment. They almost all have seizures. They almost all have ventriculomegaly, and there's other, and corpus callosum loss, volume loss, and other symptoms. So we were able to sort of now define a symptomology for individuals that share in common variations in this gene called DND5A, right? And we also got MRIs from them now. And so you can see this MRI looks actually pretty normal. There's a bit of ventriculomegaly here, but this one looks pretty normal. And that one, not so much. And here, and here, and here, and here. To this patient where almost their entire brain is just an incredibly large swollen ventricle. And they have a range of symptoms. So the idea here is, what can we do to help these patients? We probably can't, it's going to be very difficult to treat them. But there's genetic counseling. There are certain kinds of behavioral interventions that can be done. And also just for the parents, I talked to two of the mothers of two of these children. And when they just found out, because they've been wondering their whole lives what was wrong ever since their children were born. And I talked to two mothers and I told them, and I told them what had happened, and as best I could. And I tried to be as honest as I could with them. They were so relieved. They were so happy just that there was somebody out there trying to figure out what was going on, and that, that there was an answer, it wasn't anything they did wrong. Okay. And so that was something I think that is important for the families. And to study this better, what we've done is we've made a mouse model. So basically we've introduced into a mouse the same genetic mutation that we find in one of the patients. And we've worked with this guy, David Rutko at the Montreal Neurological Institute at McGill. And so this is Emily's paper. So here's 3 mice, and here's the MRI of the mice. Here's the brain here in the mouse. This is all the skull, but here's the brain. Maybe I hope you could see the ventricles here and here and here. And that's the wild type. These are wild type mice. And here's the mutant mice. And so David actually puts the mice into an MRI. This is called a 7 Tesla small animal MRI. It's got a very small bore, very powerful MRI, but it's only for research purposes with rodents and small animals. And so the mouse recapitulates at least the ventriculomegaly. So it looks like it's a good model. So now we have a way to study the details of what's going on in these brains by using a mouse model. Okay. The other way we do this now too, which I'm not going to show you, is we can get samples from the patients, which we've done, stems of just regular skin cells. Usually it's a fibroblast. It's a biopsy. It's a very tiny biopsy from the skin. You can actually use cells. You can actually take urine, and the urine sloughs off epithelial cells from the bladder. And you can take those cells and you can convert them into something called induced pluripotent stem cells. So you can convert the adult, or at least the mature, the non-embryonic cell, a fibroblast or an epithelial cell, into a stem cell. And then you can reconvert that stem cell back into a neuron. So we can study neurons from the individual patients that have the mutations, right? So that we're doing, those are the two approaches, are the iPSCs and the mouse model. So to summarize this part of the talk, the brain is complex, allowing for its amazing capacities. The development of the brain is complex, of course. ND5A is a critical component in brain development. And we can define a brain disease with implications for treatment, for counseling, and for just the emotional well-being of the parents. So I'm going to do the next part, and I'm not sure my timing here because I've lost, what time is it? Pardon me, it's 45? 35. Oh, very good. Thank you. That's good. Okay, this is going to be about 10 more minutes or 15 more minutes. Okay, so the title of the talk, my first, it wasn't that clear because I have a second thing I want to tell you about, which is only sort of partially related. It's more of a tool related part to the talk. And so the making of an unhealthy brain and the tools required to study that. And it turns out in cell biology and in neuroscience, the tools are critical. Tools actually often drive the science. You know, sequencing approaches, the development of iPSCs, you know, CRISPR-Cas9 as a way to modify the genome of cells. These tools are often leading the biology. Okay. So there is one tool that's incredibly important. They're called antibodies. Okay. So this part of the talk is now going to be a switch, but I think it's going to relate back to the first part. And I hope I can convince you that. So DNA provides the blueprints for the 20,000 human genes, for the 20,000 human proteins in the genome, and understanding what the proteins do is essential for curing disease. I think I've just given you an example. I don't know if we're curing the disease, but we certainly are understanding the problem. And the way a cell biologist and a neuroscientist studies the proteins, one of the crucial tools are antibodies, okay? So antibodies can be or are highly specific, okay? And the one example I think everyone in this room now knows about is you get immunized with either a protein or a messenger RNA that makes the protein for the spike protein of SARS-CoV-2, and you make an antibody against that. And hopefully your immune system makes an antibody against that protein and against no other protein. So your immune system is able to be very selective. So if you do that now, if I inject an animal with ND5A, an experimental animal, the animal can make an antibody against ND5A that's specific to ND5A, and we can study that protein. Okay, so what happens though now, we don't make antibodies much anymore, we buy them. Okay, so scientists purchase antibodies from manufacturers. So unfortunately, so we buy them, we pay a lot of money for these things. And it turns out, and about half of those commercial antibodies don't actually work, or they don't perform as they're supposed to. And if anyone in the room here, and I know some of you use antibodies in your research, every single scientist who does any kind of biomedical research involving antibodies has come across this problem. Antibodies that do not perform as described. It's been a problem for about 30 years. most antibodies have never been carefully tested, and it's a driver of something called the reproducibility crisis. Now, that's a bit over the top. Okay, but there are certain problems in reproducibility where pharma takes academic research and tries to turn it into drugs. And so they start by saying, can we reproduce the academic study? And unfortunately, in too many cases, either bits of that academic study or the entire thing can't be reproduced. And so pharma wastes a lot of time using academic research as a hypothesis for their drug discovery. And you know, it's flawed. And one of the major reasons it's flawed is because of antibodies. Okay, so I'm going to give you one example. And this is from my own lab. This is work that we published a couple of years ago on amyotrophic lateral sclerosis. So my lab has a major program on studying ALS and trying to understand what's going on in ALS. DND5A thing is a neurodevelopmental. This is neurodegenerative. This is something that develops when you're an adult. It's a devastating neurodegenerative disease. 90% of the cases are sporadic, 10% genetic. And it's a loss of those motor neurons, you know, the thing where the cell body's here in the room and the synapses in Saskatchewan. You know, it's amazing that those neurons are functional and can maintain the physiology they need to keep that neuron alive. And often they die, not often, but they can die. And it's the loss of those motor neurons that is what causes ALS. And it turns out that the genetics and the pathophysiology have recently shown that ALS is absolutely linked to another disease called frontotemporal dementia. So in the Montreal Neurological Institute, we study ALS. We have an outpatient clinic for ALS. But patients with frontotemporal dementia go to the Douglas halfway across the city because they have dementia. Okay, but it's the same damn disease. It's just a different neuronal population. They should not be seen at different places. Okay, and so this is what we need to understand. And one of the ways we understand that is through antibodies. And a few years ago, this major ALS frontotemporal dementia disease gene was discovered called C9orf72. Tons of papers about C9orf72. It's the major cause. So in certain patient populations in Finland, 20% of every patient that has ALS has a mutation in C9orf72. Okay, and in North America, not just 20% of those with the genetic cause of ALS, 20% of every ALS patient, and it's probably even higher. So it's really important to understand what the C9orf72 protein does, right? So it encodes a protein of unknown function. We got interested in it because the protein itself has a DEN domain, that same protein module that DND5A had. My lab had discovered DEN domain proteins as being important in membrane trafficking. So we said, oh my goodness, here's this major ALS gene, it's got a DEN domain. It almost certainly involved in membrane trafficking. So back to that same picture I just showed you. So we wanted to figure out, as I said, what does a cell biologist do when they want to understand the function of a protein? They want to figure out where it's localized. That's step number one. So there'd been lots of papers. This is an important thing. People are dying of ALS. So we went to the literature and we said, okay, where is this C9orf72 expressed in the neuron? So we found a paper that said it was expressed in the nucleus. And then we found a paper that it was expressed in endosomes. And then we found a paper that said it was expressed in the Golgi and in autophagosomes. And then we found a paper that said it was in the cytosol. And then we found a paper that said it was expressed on actin. Okay, there's just no way. No way. Something's wrong here, right? It could be. Doc back in the back there, my former mentor, knows that that's probably unlikely. So why is there a lack of consensus on C9orf72 localization of function? We think it stems from the use of non-specific antibodies. It's estimated that there are between 3.5 and 4.7 million commercial antibodies in the freezers of antibody manufacturers and distributors around the world, right? We don't need to make antibodies anymore. We need to validate the ones that are out there, okay? So we set off on a problem. This is Carl Laflamme. He's a great postdoc, as you can see. Studying antibodies has affected his status here, but okay. So, Yeah, he's a great guy. Runs my lab to a large extent. So here's an imaginary Western blot. Okay, I've already shown you some Western blot, but here's an imaginary one. This is the prototypical one. If they all look like this, we'd all be happy. So here's the blot. So at the bottom is what we've done in this imaginary thing is we've taken a cell line where we have the wild type cells, and then we've knocked out. KO stands for knockout. And we've knocked out, and I'm sorry it's a little small, but you'll see why in a second. So we've knocked out one gene. So if we look at the distribution of the proteins, so this dye just stains proteins indiscriminately, right? And so they look exactly the same because there's only one gene missing here that's present here, okay? And so if the antibody is specific, it'll recognize one band, okay? And we knock it out using CRISPR-Cas9. It's really a remarkable technology that's been widely used now. So we knock it out. So you'll have one band that goes away in the knockout, okay? So what we said, okay, if all these, if C9orf72 is located in all these cellular locations, something's wrong here, okay? So why don't we just purchase every single antibody we can purchase from every supplier in the world, make a wild type cell and another cell where we knock out C9orf72 selectively and screen through those antibodies, okay? So here's what we see. So look at this one. This is from Abcam, one of the companies. Look, there's a band. And it goes down in the heterozygote. Don't worry about that too much. But there's knockout A and knockout B. We made two distinct lines to make sure we were being specific here. And you see, get my mouse back here. Where'd it go? There it is. And see, the band's gone. But this one, I want to come down to this one. Look at this one. There's a band exactly the right size. This is from a company called Santa Cruz, and any of the cell biologists or biochemists in here will nod their head about that one. Notoriously bad antibody company. So here's a band, and look what happens in the knockout. It doesn't go away. That's not C9orf72, but it's exactly the right size. And then what we found was this and this. So let me just show you. Here's an antibody from Abcam. Beautiful. Here's an antibody from GeneTex. Beautiful. Here's the antibody from Santa Cruz. Not beautiful. Okay. I'm going to skip through this slide. I think this is just a little bit. This is immunofluorescence. It just showed that the same Santa Cruz antibody didn't work in immunofluorescence, but the Abcam antibody did work, or one of the GeneTex antibodies did work. So monoclonal antibodies against GeneTex and Abcam were optimal for immunoblot. There was another antibody from GeneTex that's optimal for immunocytochemistry. At the time, neither of these antibodies had ever been used in a published paper. The Santa Cruz antibody had been used in 16 papers, and those 16 primary papers had been cited 3,700 times. Okay, so bad science being propagated. And what happens is the first paper uses the Santa Cruz antibody, then the next paper says, oh, well, they used the Santa Cruz antibody in their paper, so we'll use it again. And then the third paper is, well, these first two papers use the Santa Cruz antibody. And that antibody simply does not recognize the protein in any application, period, right? So there's a problem. So why are the companies selling bad antibodies? And why are the scientists buying them and using them? Right? That's a double problem here. And it's not just a science problem, it's also a business problem. Okay. So it's expensive to test antibodies. It's really not cost effective for the companies. The manufacturers kind of say, well, the scientists should be testing them. The scientists say, well, we paid for them, you should be testing them. The granting agencies and the journals want validated antibodies, but they don't demand it. They don't force it. So the scientists say, well, we don't have the money and testing's expensive, and some of us don't even know how to test properly. And these are not trivial things we're doing here. We just do them a lot in my lab. And it's not required, so why do we do it? So it's estimated that about $1 billion annually is wasted on bad antibodies, just in North America, okay? And that propagates bad science. So we've come up with a solution. It's a little bold of us to think we can solve this problem, but we do. We think we can solve this problem, and we think we have a great solution. It's based on the scientific principles that Carl did in his eLife paper, as using the C9orf72, where you do a wild type in a knockout cell, and you get every antibody from every company. So what we've done is we've created a new, what I would call concept for antibody characterization testing called YCAROS. So that stands for antibody characterization through open science. So we envision Ycaros kind of like a bond rating agency, like Moody's or Standard & Poor's, or a product rating agency, or maybe best, a product safety organization. And Ycaros is an incorporated company, okay? The company currently has one part-time employee who's the CEO, but all the academic work, all the testing is being done in my academic lab at McGill. The idea is to eventually transfer that to Ycaros or some other organization. Okay, so unlike most companies, Ycaros is very different. Okay, so the way it works is a funder nominates protein targets. So, for example, the National Institutes of Health in the U.S. has given us a grant for $1.6 million US dollars to study, to validate antibodies against Alzheimer's disease targets that have come from their research, okay? ALS charities in Canada, the United States, and England have nominated ALS targets. So and then what we do is we collect all of the antibodies from all of the antibody manufacturers in the world. But I'm going to show you in the next slide, uniquely what we do is we don't buy them anymore like we used to. We've brought all of these antibody manufacturers on board to our Ycaros concept. They provide all of these antibodies as in-kind contributions. And I'm going to give you just a visual example of that. We test them using the procedures I outlined. We provide open feedback to the companies, and every piece of data is put into an open science repository without restriction. Okay, there's no intellectual property, no patents, no investors, no financial conflicts. I often question why the hell I'm doing this and spending all this time doing this when I'm not even an investor. All data placed into public databases. It's a public good. Okay, we think this is a problem and we think we can solve it. So here's the solution. This is the Ycaros shown as now in a graphic form. So these are all of the funders that we've got on board with us. ALS societies, Roche, one of the major pharmaceutical companies, the NIH, Resolute, which works on transporters, Genome Quebec, Genome Canada, and others. These agencies give us money to the lab. They nominate targets that they want studied, and then we go to our manufacturing partners. These partners now represent about 70% of all antibody manufacturing capability in the world. Okay, and they are on board with this completely. We have monthly meetings with our board of directors. Every one of these companies has a representative on that board. We give them the target, they go through their catalogs, they find their antibodies, they ship them to us, they pay the shipping charges, we get the antibodies and we do the validation in the lab, okay? And two of these companies actually provide us with knockout cell lines. Between them, they have about 10,000 different knockout cell lines. If we don't have the appropriate knockout line, we make it ourselves, or we make it with a collaborator, okay? Then all of the data is placed onto Zenodo, an open science data sharing site. So up to now, we've published about 55 reports. We put these all into public databases. We've tested about 750 antibodies. We've had in-kind contributions, the antibodies and the cell lines of about 1.3 million. We've had cash contributions of over $3 million to this project. And now what we're trying to figure out is how many antibodies actually work. About 64, and work, that's a tough word, comparing IF to IP to a Western blot. If you want to make a bold statement, about 2/3 of every commercial antibody does not recognize the protein that it's intended to recognize. Okay, and so we're trying to go, and what's happened actually, which is beautiful, is that the companies, as we feed that back to the companies, when an antibody works, they're able to cut and paste out of our public report, put it onto their own data sites and use it as marketing material. Okay. And when they don't work, we've had about 25 examples now where companies have pulled the antibody off the market. Okay, no longer on their website, no more advertising, gone. So we're starting to make a difference. And our goal is in the next 10 years to do this for 20,000 human proteins. And we're building towards that. And that's a very difficult thing to do in the confines of an academic environment. We're trying to figure out mechanisms, and we have some. So I think in conclusions, we can solve the antibody problem with an open science public-private partnership. And the open science is key to this. If it wasn't open, these companies would not stay on board with us. We'll do better science and contribute to understanding neurological disease. Second last conclusion, I have the best job in the world. And the last conclusion is science really does matter. Okay, so this is our group. This is the Ycaros group. And it doesn't, Wykeros is in my lab. So this is my lab, Wykeros is my lab right now. Like there's no separating them. But so Chaitan Rainen, our CEO, this is Al Edwards, who's the head of the Structural Genomics Consortium at the University of Toronto and also professor at the University of Toronto. The science was me and the business model was Al. It's a beautiful business model. And Al is he's a remarkable guy. Carl Laflamme is a postdoc research associate in my lab who's been driving this. And over the years, I've been so fortunate to have so many great people in my lab. And, I was trying to get at the beautiful thing is, I get older and older. In every picture, I'm older and older and older and older, but the people in the lab are all the same age, right? I'm thinking, how is this going on? What's happening here? So it's just so great that I get to work with these young, energetic people in my lab. So thank you very much for your attention.
But let's get some questions going. We're going to go. So one of our virtual participants, the question is, there's a lot of mass media about plasticity and rewiring of the brain. Please speak about the neurobiology mechanisms on how we can train/nourish the brain to escape from unhealthy psychological habits. Great question.
A very complicated
Yeah.
question. That's about 3 or 4 questions, I would say, actually. To escape from unhealthy psychological, well, so let's start with what I'm showing you, that DND5A epileptic encephalopathy. That's an unhealthy brain. So that's an unhealthy brain where the development of the brain is so clearly abnormal. You can't train that brain, unfortunately. You can't make that brain through synaptic plasticity changes a more functional brain. Those individuals are so severely disabled. But if we want to take it to another end, where we have individuals with psychiatric disorders, with neuropsychological disorders. There's lots of examples of neuropsychological disorders, anxiety disorders, post-traumatic stress syndrome, bipolar disorders, where behavioral therapy, cognitive behavioral therapy works very, very well. You can train your brain. And what you're doing when you're training your brain like that through those kinds of cognitive therapies is you're actually rewiring your brain. You're actually changing everybody who walked in this room today will leave and their brain is different when they walk out the door. Most of you have never seen me before. You'll walk out the door and now you'll have an image in your brain. It'll go away over time, probably pretty quickly. It scares you. So your brain has changed in that time. Your brain has physically changed. You've rewired your brain because you now have a new memory that wasn't there before. So in between that are the things like ALS and Parkinson's disease, where maybe there are, motor neuron, spinal cord injuries, right? Can we make the neurons regrow? Can we make the neurons, that have been cut, can we convince them to regrow and reinnervate their target properly, right? So there's a whole range of things in there. But we can certainly, I think the last part of that question was unhealthy habits or unhealthy psychology. That we can do, that commitment from an individual with one of those neurological or neuropsychiatric or psychological disorders. Cognitive behavioral therapy is one example that works beautifully.
Thank you. I'm gonna bring the mic to you just because otherwise our participants virtually won't be able to hear.
Thank you. Really enjoyed the talk.
Thank you.
Evolution has built-in redundancy to so many physiological systems. It seems to me that your example of DND5A, there is no redundancy.
Yeah.
What happened?
Yeah. So the remarkable thing about this is that there's another gene called DND5B. It's about 90% identical. It's localized to if you do co-localization of DND5A and DND5B, they co-localize perfectly. They probably have the same membrane trafficking properties. DND5B is not affected at all in these patients. This is a thing with a beautiful redundant mechanism built right in there. There's no variance in DND5B and there's no change in the expression level of DND5B. Why isn't DND5B compensating, right? Some genes, C9orf72, that mutation, there's only one mutation in C9orf72 and it's expansion mutation. And if you have that expansion mutation, the penetrance of that mutation is basically 100%. You are going to get you're either going to get LS or frontal temporal dementia or both. One would think there's other D-domain proteins, there's other proteins involved in doing the trafficking functions that C9orf72 is doing. So the DND5A is not an uncommon example. That is all too common. You know, and the parents of those patients are heterozygotes. They have half of the protein. We're not sure if it's a loss of function or a dominant gain of function mutation, but and they're fine. They're perfectly fine. So, you know, what we believe the protein is doing, what we really believe DND5A is doing, is it's actually part of something called the Crumbs complex for the aficionados. So during neuronal development, as the neuronal precursors divide, they undergo rounds of cell division, and you make more and more precursor cells. But at some point, they undergo an asymmetric cell division, and they go to a precursor and then on a pathway to a mature neuron. And we think what happens is that this DND5A is really important for this Crumbs complex. So it's all about cell polarity. So when you disrupt DND5A, you're unable to undergo these cycles of polarity, loss of polarity, polarity, loss of polarity as the cells undergo these cycles of division. And so what happens is they fall out of the symmetric cell division too early. They go into an asymmetric cell division. You don't actually have enough stem cells, enough neuronal precursor cells. The individuals don't have enough precursor cells to create enough neurons, and the neurons that are made differentiate too early, don't know where to go, and go an apoptotic cell death. So it's a double whammy in that respect. But why is there no compensation? You know, these are complex questions. Yes.
So what? They won't be able to hear.
That's for the curious. People with frontal temporal dementia don't necessarily have symptoms of,
Motor.
of ALS, and
Yeah.
people with ALS don't typically have symptoms of frontal temporal dementia, from what
Yeah.
I've seen.
Yeah. So this was, so it's a great point, right? And again, another one of these mysteries, right? And this is why I kind of started by saying, we know a lot about the brain, but nobody, I don't believe in the world, certainly not me, knows how brain turns into cognitive ability function. Okay. So really, and as I say, so in Montreal, the Douglas Hospital saw all the dementia patients, the FTD patients, the neuro saw all the LS patients. These were different diseases, completely different. Never the twain did meet. But so when C9orf72 was discovered, it turned out that patients, that was kind of the first clue, was in 2011 with the discovery of C9orf72, that half the patients some percent of the patients would develop with the C9 mutation would develop the frontal temporal dementia, some ALS, and some have both symptoms. And so absolutely there is frontal temporal dementia patients with ALS-like symptoms and vice versa. Okay. But it's still uncommon, and it's mostly happens with the C9orf72 mutations. So as far as we can tell, C9orf72 is expressed in the spinal cord, it's expressed well in the cortex. So why the brother and the sister, or the brother and the brother, or the sister and the sister in these large families, and a lot of this comes from these Quebec families that our director Guy Rouleau has studied. One sibling develops a pure ALS, one sibling develops a pure frontal temporal dementia, and the other develops a complete hybrid of the disease, of the two diseases. So they're not two diseases, actually. It's just they're different neuronal populations with the same pathophysiological mechanism, we think. Okay. So why? I think that's something we'd like to understand. That's why we really want to understand the function of the protein at the cell biological level, and what's going on in a motor neuron versus a cortical neuron.
Could the combo just be misdiagnosed?
It could be, I think, because again, this point, right? It's psychiatrists dealing with the frontal temporal, and it's neurologists dealing with the ALS patients, right? So they probably a lot of times they just miss it. Yeah. But probably most ALF patients have some dementia towards the end. Most frontal temporal dementia, it's in Parkinson's, for example. All the Parkinson's patients start with a tremor, but they all, by the time the disease is running its course, they're dementia patients, right? And so it might just be a question of timing. and then there's, the other big consideration in all of this, which is kind of, again, for the aficionados, is the immune system and immunological responses. So my colleague Heidi McBride at McGill had, so patients who have a Parkinson's disease mutation called PINK1, it's 100% penetrance. It's a rare mutation in Parkinson's, 100% penetrance, right? Without fail, they develop Parkinson's disease, and it's a juvenile onset. They get Parkinson's at the age of 30, okay? You make that same mutation in mice, they never get Parkinson's disease, ever. They never get anything, they never have a symptom. What Heidi does is she takes them, she gavages them one time with a gram-negative bacteria, gives them an immune challenge. Normally, the mice are living in a HEPA-filtered cage. They never have any immunological challenge. Now the mice all develop Parkinson's-like disease, right? So the humans are exposed to those immunological challenges all the time, right? So so some of these things may be related to an immune challenge in the environment that's far beyond the genetics.
There's a question from our virtual audience, and that is, drugs or digital addictions impact the brain. So can we start to find a way to understand how the brain is damaged?
Again, if you're it's the same kind of question as the first one, right? Drugs or digital addictions. I mean, so all of those, so drugs, gambling, eating, sugar, drugs of abuse, alcohol, a lot of them hit a common dopaminergic pathway. That pathway makes you feel good, okay? Making you feel good makes you want to feel good again. It wants to reinforce feeling good. So impact the brain, so we can start how it's damaged. I wouldn't say that the brain is damaged from addiction. I wouldn't say that the brain is damaged. In fact, it may be that certain damage to the brain is what drives the addiction. So smokers, it turns out that for smokers, they actually have pretty low, you can look at autopsy and find out that the smokers had lower levels of certain dopaminergic receptors than non-smokers, okay? Did the smoking cause those dopaminergic receptors to downregulate, or was it the fact they had a polymorphism, just normal variation in every human being, so they had lower levels of the dopamine receptor to begin with, so they smoked to make up for that, to drive dopamine expression in the synapses, right? So but I wouldn't say that the brain is, that those things cause damage to the brain, but they certainly change the brain neurochemistry. And yeah, again, counseling, cognitive behavior therapies, and certain drugs that can go in there and not fix the brain in terms of it being damaged, but certainly fix the behavior. But again, this is the tough line. What is a damaged brain versus a behavior? An individual I know has a disease called fibromyalgia, okay? She suffers from pain. This is the main symptom, pain and fatigue. But there's no problem. There's no chemical issue. There's no genes described. There's nothing wrong on the MRI. And everyone says, well, the pain's in your head. Well, where the hell else is pain if it's not in your brain? Right? And so she's told she's got a psychiatric condition, a psychological condition. Okay, but if she's suffering from pain, she's suffering from pain. The pain's real, right? So what's the difference between a psychological and a neurological condition? In my opinion, there should be psychology and neurology. And psychiatry, I think, as a discipline should be eliminated. I think psychology is very good at helping people understand the issue and training their brains to overcome the mind issues. Neurologists are good at understanding the function of the brain. Psychiatrists are somewhere in between, and I just don't think psychiatry is a discipline that should exist. I hope there's no psychiatrists on the line.
Last question over to you.
Yeah.
Thank you. Fantastic talk.
Thank you.
I really admire the work you're doing with YHROS, because I too believe that bad antibodies have created a lot of problems in scientific discovery.
Yeah.
I'm a little disappointed to see that the CIHR wasn't somebody listed on your funding. Have you reached out to them? I would hope that they would fund this because it's really important.
No, we haven't. Right now, we haven't needed to. I think we got 1.6 million from the NIH and we got 4 million from Genome Canada. CHR, as you know, wants a hypothesis. It's their hypothesis-driven research. The hypothesis is antibodies are bad and we're going to correct it. It's just not. And the NIH grant was actually not a grant. It was a research contract from an existing grant at Emory University. And so they had a grant on Alzheimer's disease. They were discovering these dark Alzheimer's disease targets, and they contracted us more, right? So CHR, I think, would not be very interested in this, and because it's not a hypothesis-driven thing. Actually, the one area we're really focused on, and we're still hoping, is something called FROs. They're called Focused Research Organizations. And there is a company called Convergent. Again, it's a non-for-profit, open science kind of corporate group. And it's within the Schmidt Family Foundation. This is Eric Schmidt, the original CEO of Google. And what they do is they look at projects that are sort of academic projects that should be in a biotechnology sector, but don't have the right profile. We're never going to get venture capital to support this, right? But what about philanthropic capital? So they basically have a mechanism where they fund you at the $50 to $75 million range over 5 years. But more importantly, well, 50 to 75 million is pretty important, but more importantly, what they do is they set you up. So they get you the research space. They hire the executives. They deal with all the compliance, with the tax implications, with the shipping, with the HR. They've got experts to do that. You just then move the idea into that structure. So it runs as a biotech company, but based fully on philanthropic capital. And we've been in serious discussions with them. So that's, I think, a better way to go about it.
Okay, I think we're over time. So
Okay.
thank you very much,
Very
Dr.
good.
McPherson.
Thank you.